We are about to start the next session. My name is Karel ten Katen. I will be chairing this session. It's a great pleasure to introduce to you Debby Kelly from University of Manitoba, where she is professor in psychology and adjunct professor, whatever that might be, in biological sciences. And she also has a research chair over there. So her work is also on spatial cognition. Like we heard a previous talk with a wonderful work done on bats and how they orient in space and how they can make objects and things like that. Her work is also on spatial cognition, but on birds. And very interesting work which is being done on what exactly do birds remember about the space around them, how they hide food, how do they find their way through space, and what sort of features do they use of their environment to get anywhere. So although her current work is in birds, she has worked on quite a few other species. So she is really a comparative cognitive scientist. And she has also been chair of the Comparative Cognitive Society. So I'm very happy that you're here to give this talk and looking forward to what you have to say. Floor is yours.
Thank you very much. I'd like to start by thanking the organizers for inviting me here today, especially Stephen. Thank you so much. It's quite an honor listening to the talks online and being here in person to hear the talks, to think that I was asked to be involved in this really wonderful 2-week workshop with some really amazing people and amazing talks. So thank you very much. So I'm honored to be able to talk about one line of research that my students and I are working on in my lab at the University of Manitoba. And I titled my talk Spatial Cognition in Food Storing Birds. And then thinking about it, it's really cognition in food storing birds that I'm going to talk to you about. And I'm going to weave spatial cognition through that theme. So many researchers who are interested in spatial cognition or in comparative cognition are really trying to understand how animals interpret, maybe think about or reason about their environment. And we can come up with hundreds, thousands of examples of the types of questions that researchers ask when they're asking these comparative cognition type of questions. But I'm not going to belabor you with hundreds and thousands of examples. I'm just going to come up with a few. So we can ask questions about whether animals are able to engage in thinking about their past, perhaps planning for the future, episodic memory or episodic-like memory. We can ask about how they engage in navigational feats. Do they encode routes? Do they plan those routes in advance? We can also look at whether animals are able to reason about their environment in terms of wants, needs, some people say desires, about other individuals within their environment, and those could be conspecifics or they could be heterospecifics. One of the main goals when we look at comparative cognition and researchers studying comparative cognition is to try to better understand how cognition has come to evolve. And perhaps it's not surprising then to know that there's a plethora of studies that have focused on our closest living ancestors, the great apes. And we've learned a considerable amount of information about how these animals encode their environment and learn about their environment. And as I mentioned before, there's many examples. Well, there's many examples when we look at non-human primates as well, looking at their cognitive and learning abilities. But one example, which many of you I'm sure know about, is non-human primates' ability to use and manufacture tools. So 2 examples that I'm showing here is being able to manufacture twigs to be able to get out grubs and termites from wood structures, or to be able to gather rocks and use them for nut cracking. And those are just two examples. But recently, these kinds of cognitive abilities, some call it complex cognition, some call it higher order cognition, have been found in birds. And just to parallel what I showed you with non-human primates, there's been researchers that have shown that birds, primarily in the corvids, are able to do similar types of things. So they can use and they can manufacture tools. So here's a video because I love watching animals behave, and I think it's so much more interesting than having me describe it to you. And it's a great video provided by a wonderful colleague of mine, Alex Kasalnik, and his colleagues showing tool use in a species of crow. So I'm going to try to walk you through this, but what you see is there's horizontal tubes, and the bird needs to get food that's hidden in one of the horizontal tubes using these tools. So the first tool is too short to be able to get at the food, but it's long enough to use as a tool to get a second tool. Once she has that second tool, it also is too short in order to be able to access the food in this horizontal tube, but it's long enough to get a third tool. When she has this third tool, that's sufficiently long enough in order to get the food reward at the back of this tube. So I think it's a really nice example of tool use in these corvids. And here's a nice representation, just generally summarizing the work that has been done looking at complex cognition in non-human primates and in primarily corvids again. And Emery and Clayton and their colleagues have categorized it into these four main types of complex cognition. So you can look at causal reasoning, imagination, prospection, and flexibility. And as you can imagine, there's been controversy about whether animals are capable of these kinds of complex cognitions and how should we really define them. But I think this is a nice representation to show the different tasks that have been used in primates as well as in birds. Well, what I've told you so far should cause some kind of concern if you know the term bird brain the way I grew up knowing the term bird brain, and if you've ever been called a bird brain or if you've ever called someone a bird brain, you've meant it in a very insulting way. But how does that fit with what I've just shown you about birds being able to use tools, for instance, or show complex cognitive abilities that rival non-human primates? And I'm just going to show you this as a bit of an overview to set the stage for the work that I'm going to be talking about. So Jarvis and many colleagues worked hard on trying to understand the neural architecture that represents or that supports complex cognitive capabilities. And this is a picture from their 2005 publication. And what's important here is just really looking at the shading differences. So we have a representation of a songbird brain and a representation of a mammalian brain, in this case, a human brain, and then a color coding, which indicates if you see areas that are shaded in green, these are areas that are primarily responsible for complex cognitive capabilities. Whereas the purple shading is areas that are primarily responsible for more instinctive behavior. And this is the traditional view or the classic view of how we used to think about bird brains. You can see the majority of the bird brain is shaded in purple. So it's really thought that much of the bird brain can only support this instinctive behavior, hence the insult. Whereas if we look at the human brain, we can see that it's encapsulated in all this green shading. So it has the ability to support complex cognitive function. However, in this process around 2005 and earlier, many important neuroscientists in particular got together and looked at the disjunct between what was known in the literature, what findings they were getting, and as well as the behavioral data and cognitive data that were coming out of research labs. And together, they made this important advance in understanding avian neuroscience as well as cognition and behavior. And that's represented in this updated schematic. So we can see now the same scale where green is showing areas that are more supportive of complex cognitive abilities, that we understand that the avian brain is actually able to engage or support these kinds of complex activities that we've seen birds doing. So with that as a bit of a foundation, I'm going to leap off and start talking more about the work that I'm doing with my students in the lab at the University of Manitoba. So many of you probably know about food storing. So there's many animals that engage in food caching behavior. And what they do is they'll go around and they'll find food during times of plenty. So typically this is late summer and autumn. They gather these items and then they put them in individual caches. So these are what are called scatter hoarding in species. And they put them in different locations around the environment such that when winter comes and resources are not as plentiful, they can fall back on these hidden caches. So to be an evolutionary stable strategy, many things must happen, but I'm going to focus on two in particular. And one is that the bird needs to be able to recover its caches, and it may do so by protecting what others know about those caches. So it may need to be able to protect those caches from potential thieves or pilferers. And research has shown that many caching individuals or many caching species indeed show cache protection strategies. So they'll hide their caches out of sight of other individuals. They'll make false caches where they'll cache inedible items such as twigs or stones. Or they'll even go back and recache their caches when they've been observed by another individual. The other thing that needs to happen is that the animals need to be able to remember where they put their caches. So there's no sense spending all this time and energy making hundreds or thousands of caches if you can't remember where they are. So I'm going to step through these two aspects as my two main themes in my talk. I have three, and the third one will come later. So let's get going and start looking at, do food storing birds in particular show cache protection strategies? And much of this work started with Emery and Clayton and their colleagues, and they've primarily looked at scrub jays. They've looked at other species as well, but primarily looked at scrub jays. In my lab, we're looking at Clark's nutcrackers and pinyon jays, and we've chosen these two species because Clark's nutcrackers are highly spatially dependent, so they'll store about 30,000 seeds in the fall and remember where those caches are throughout winter. But they're fairly less social species, so they'll form pair bonds or they'll form mating pairs, but they're quite territorial. Whereas pinyon jays also are highly spatially dependent, so they'll make about 20,000 caches, so fairly similar. But they're very social. So they'll live in flocks of sometimes hundreds of individuals that are typically closely related. So this gives us two ends of the spectrum, if you will, or two points along the spectrum to understand whether sociality plays any kind of role in whether birds engage in cache protection strategies. And if they do are those strategies same or different? All right, so let's get started. As I said, the procedure was first developed by Clayton, Emery, and their colleagues. And I'm not going to walk you through what they've done because we tried to replicate it as close as possible. So to save a little bit of time, I'll show you our procedures. They're quite general. So we bring in wild-caught birds into the lab. We give them a bowl of pinyon pine nuts in this case, and they can eat as many of those pine nuts as they want. And when they're satiated, they will just naturally start caching the remaining foods in these ice cube trays that are filled with sand. So that's the general procedure. And here's the first study I want to talk to you about. It was conducted by my PhD student at the time, Dawson Clary. And the study, I'm going to go through the general procedure. So it occurs in 3 phases. The birds are given a caching phase. So we're going to refer to this bird as the caching bird. It's given the 50 pinyon pine nuts and the 2 visually distinctive trays that you'll see in the cage here. And it's allowed to eat or cache as many of those seeds as it wants. And again, it's important to keep in mind that these 2 trays, although they look quite similar to, from this point of view, they're visually distinct. So they have different colored objects attached to them. So the bird caches, and then it moves to a pilfering phase. And in this example, the trays are put on a cart that's outside of the bird's cage. And this isn't much of a pilfering phase in that in this example, the trays are just sitting there, but that's going to be important later on in the talk. So during this pilfering phase, something happens to those trays. And then finally, at a later point in time, those trays are given back to that caching individual. And the caching individual can do as it chooses. It can take the seeds out of those trays and eat them. It can take the seeds out and it can move them to different locations, so what we call recaches. And we record the behavior during these 3 phases. Okay, so to get into the details of this study, we were interested in asking whether Clark's nutcrackers, again, these more social corvid species, does it engage in cache protection strategies? And we had 3 conditions, a private condition and an observed condition, which is very similar to what the previous studies have had, as well as an object condition. I'm going to walk you through these. And importantly, the birds received 3 replicates of each of these conditions. So they received each condition 3 times. We had 6 birds, 3 males, and 3 females. So I'm going to walk you through each condition. In the private condition during the caching phase, the bird is given those 2 ice cube trays and 50 pinyon pine nuts. They're allowed to eat as many seeds as they want, and then cache as many seeds as they want as well in those 2 distinctive trays. Importantly, there's another cage behind them, but there's a visual barrier between these 2 cages. Then in the pilfering phase, the 2 trays are just put on a cart outside that caching bird's cage, and absolutely nothing happens to them. They just sit there and the bird can see that nothing is happening to them. And then finally in the retrieval phase, oh, there's a question.
Oh, what is it?
So the question is, what does pilfering mean? Pilfering means to steal the caches. So a bird is pilfering a cache if it steals the seeds out of that caching tray. Thanks for the question. So then in the retrieving phase, those 2 trays are given back to the caching individual. And again, that individual can do whatever they want. They can eat the seeds, they can recache them, whatever. That's the private condition. The observed condition, now you'll see that that barrier is removed between the 2 caches. So the caching bird makes its caches in the 2 visually distinctive trays, but now there's another nutcracker that's watching the caching event occur. During the pilfer phase, 1 unique tray, which we're going to call the safe tray, is put on the table outside of the bird's cage. Nothing happens to that tray. The other tray, however, goes into the cage with the pilfering bird, the thief. And the caching bird can only sit on and watch while that pilfering bird steals as many caches as he or she likes. And then the 2 trays are returned back to the caching bird. And again, the caching bird can retrieve those caches, can eat them, whatever. Then we have an object condition, and this is what we added that was different from previous studies. So now in the caching phase, there's an inanimate object in the cage adjoined to the caching bird's cage. And we were interested in adding this to try to understand, are the birds behaving because there's something in the other cage? And it doesn't necessarily have to be another nutcracker. In the pilfering phase, we set it up exactly the same as the observer, where the safe tray goes on the cart outside the cage, and then the pilfer tray goes in with the inanimate object. And then finally, in the retrieval phase, the two trays are returned back to the caching bird. But what's important in this control condition is, unbeknownst to this caching bird, we as experimenters pilfer from the pilfer tray, and we yoke it to the previous observer condition. So what do I mean by that? When the bird gets an observer condition, the observer might steal 15 seeds. In the next condition, when they have the object, we as experimenters will steal 15 seeds, but the bird doesn't see us do it. So this is a way that we can also control for whether differences we're seeing is because the caches are being pilfered or not, in this case by an inanimate object. Okay, so Dawson wanted to ask whether nutcrackers will modify their behavior when they're being observed. Will they show cache protection strategies? And does this behavior change when the birds are making their caches or when they're retrieving? So we'll look at the first one. And here on the y-axis, we see the number of seeds that are being cached. And on the x-axis, I'm just showing you the first two replicates because we really see behavioral changes immediately. And we're coding it by private, observed, that's being observed by another nutcracker or being observed by the object. Importantly, in the first trial, the birds have no experience that anything bad like pilfering is going to happen to their trays. So there should be no differences between these two conditions, and indeed there is not. However, after the very first experience with this pilfer event, the birds are responding differently when they're being observed by another nutcracker. They're reducing the number of caches that they're making in those trays. So what do they do when we give the trays back to them? Do they change their retrieval behavior? Here, what we're looking at is the proportion of seeds that the birds are recovering. So they're primarily eating them in our experiments. And we looked at private, observed, and object condition. And what you can see is the nutcrackers recover more of those seeds. So they eat more of those seeds when they've been previously observed by another nutcracker. And they show no differences when they're in private or being observed by an inanimate object. Okay, so we know that Clark's nutcrackers suppressed their caching behavior when they were observed, and that they recovered more caches when they were observed. And importantly, we saw no differences between the private and the object condition. So adding in our control condition allowed us to really understand whether the behavior we saw was dependent on something, perhaps an occasion setter in the next cage, or whether it was related to the reduction of caches, and this doesn't seem to be the case. Next, we want to go on and look at more social species, and this is our pinyon jay, and we're going to ask the question of, does it matter who's watching? So this is work done by my current graduate student, my current PhD student, Alize. And she basically replicated the study that I just talked to you about that Dawson conducted. So I'm going to go through it a little bit more quickly, but you'll see that there's a few important differences. So the pictures are different, but the situation is very much the same. And she's going to ask whether Clark's nutcrackers and pinyon jays modify their behavior. So Dawson already gave you a bit of foreshadowing of what we expect to see with the nutcrackers. And does it matter who's watching you when you're making these caches? And again, she's going to look at caching behavior and retrieval behavior. So same kind of setup. The birds get this caching phase. They can eat pine nuts until they're full, and then they can start caching them. They have a pilfer phase where something happens differentially between this safe tray and this pilfer tray, and then they're given both trays back to retrieve. So now we have 4 different conditions asking the same question and 3 replicates of those 4 conditions. So we have the private condition, which you've seen already. We have observed by a conspecific. So this is similar to what you just saw in Dawson's study, but now we're adding observed by a heterospecific. So if a Clark's Nutcracker is making caches, does it matter if the observer is another Clark's Nutcracker or if it's a Pinyon jay, and vice versa? If a Pinyon jay is making caches, does it matter if it's another Pinyon jay observing or if it's a Clark's Nutcracker? And then, of course, we had our object condition, and we had 10 birds in this study. So you've seen this before, but I'll make sure that you understand this. Caching phase, given pine nuts, cache as many pine nuts as you like. In the private condition, one tray is, or both trays are put in an inaccessible location, so nothing happens to them. And then retrieval phase, they simply get those trays back. When they're being observed by a conspecific, there's another, in this example, Pinyon jay in the cage next to the caching cage. It's watching as the bird is making those caches. In the pilfering phase, only the pilfer tray goes into that observing bird's cage. The safe tray remains safe outside both cages, and then the birds recover. Here's the conspecific, or the heterospecific example of a Pinyon jay being observed by a Clark's Nutcracker. Then the Clark's Nutcracker is able to pilfer as many seeds as it wants from that pilfer tray, and then the Pinyon jay receives both trays back. And the object condition, caching with an inanimate object, the object pilfers some of those caches, and then the birds can retrieve from both trays. So the same 2 points, do they change caching behavior and do they change retrieving behavior? So here, again, we have the mean number of seeds on the y-axis, and then we have our trials, our replicates, if you will, on the x-axis. And we're looking at our private object observed by a conspecific and observed by a heterospecific. And this is the data just with the Clark's Nutcrackers to start. And you can see that the Nutcrackers reduce the number of seeds that they're caching when they're observed by a conspecific, so another Clark's Nutcracker, but they don't change their behavior when they're being observed by a heterospecific, a Pinyon jay. And this doesn't change over time. So the very first time they experience it, they show this drop in caching behavior, but for the heterospecific, that doesn't change. What about the social Pinyon jay? Well, I'm gonna delay my answer to that because I think it's really important to foreshadow that there's something different happening in the safe tray and the pilfer tray. So my graphs are gonna now show you the mean number of seeds cached on the y, the trials on the x, but I'm gonna divide it out into responses or caches made to the safe tray and caches made to the pilfer tray. And importantly, the Pinyon jays are not showing differences between these 2 trays when they're caching in private and when they're caching with an object. That's an important baseline for when we move on to look at how they respond to the conspecifics and heterospecifics. So the very first time they experience a pilfering event, they show a difference in where they're putting their caches. So they're putting more caches in the safe tray and they're putting less caches in the pilfer tray when they're being observed by a heterospecific, but not when they're being observed by a conspecific. But by the second time they experience these pilfering events, they're treating them both as potential pilferers. So it's like they learn over time that, or they need a bit of time to learn that a heterospecific can also be a cacher. So what did we find? We found that Nutcrackers reduce caches when they're with a conspecific, but that Pinyon jays initially reduce caches with a conspecific and then also reduce caches with a heterospecific. So does retrieval behavior change? Well, let's look first at Nutcrackers. Remember, Dawson's work showed that they do. So, but here we see this you may want to call it, some people call it a trend. We didn't do trend analysis, so I can't tell you if it was a trend, but there's not significant differences between the conditions. So it seems like they show a bit of a difference, but these are not significant. And the pinyon jays also don't show any significant differences in the proportion of seeds that they're recovering during these 4 different conditions. Okay, so no behavioral differences between private and object conditions. We see species differences in their cache protection strategies. So nutcrackers seem to show this overall cache reduction when they're being observed by another Clark's nutcracker. They just simply reduce the number of caches they make, whereas pinyon jays show more of a selective cache reduction or a cache protection strategy, if you will. They hide more caches in that safe tray and less caches in the pilfer tray. And both species show no differences in their cache retrieval behavior in this experiment. Okay, so just to summarize this first theme, we see that food caching birds know when they're being observed and engage in cache protection strategies. And we see that these strategies differ for the Clark's nutcracker and for the pinyon jay. And these differences may reflect sociality. And I say may because, of course, we've only looked at 2 species, and there are many other alternative explanations or correlations that might go along with sociality in these cases. But I think this is a really nice first start at being able to look at whether the bird's social environment has any effect on its cache protection strategies. So the second thing that I said caching animals need to do is they need to remember where they put their food stores. So now I'm going to switch gears and look at, well, what spatial cues do birds use in order to remember their cache locations? Is there a question?
Yep. Before we get to the social cognition, are pinyon jays family groups?
Yeah, the question, do pigeon jays live in family groups? So they live in these large flocks of typically closely related individuals, yeah. And they can be up to 100 individuals in a flock.
So when you looked at the pinyon jays, did you have them as a family group?
Yeah, that's an excellent question. No, we did not. At the pinyon jays that we have, aren't necessarily all from the same family group. So all of these studies were conducted in the lab and not in the wild. It's like I said, there's a lot of future questions that need to be asked in order to find out why these birds are different. And it may be sociality, and it may be something important about that social structure itself. So you could also ask about potential, do they pilfer differently if it's a mate versus...
Yes, I would think it would make a difference. If you had kin, you would expect them to be less selective.
It might. We haven't done those empirical studies, but those are great questions, absolutely. Okay, so now switching gears to spatial cognition. So Clark's nutcrackers, food storing bird, I said they cache about 30,000 items in the fall. They scatter hoard where those caches are made over fairly large distances, and then they have to retrieve them in the fall. And so what I want you to think about is how the spatial environment changes from the fall when they're making these caches to the winter when they're having to retrieve them. So here's just a little video to show you a Clark's nutcracker making a cache in the fall. So it's taking pinyon pine nuts that it's storing in its sublingual pouch and sticking them in the ground. And so even though this bird has taken 2 or 3 seeds and stuck them in the ground, that's considered one cache. So I'm just going to play it again. Have a look at the spatial cues that you might use if you were trying to relocate that cache. It's fall or autumn, so there's dead grass around, there's, a big rock in the scene. Those are the kinds of local cues that are around that cache. Now it could be 6 months later, and the bird needs to remember where it put that cache. And you can see that the spatial environment has changed considerably but the bird is still able to find its cache. So a lot of those local cues, like the grass that you saw, are obliterated by this thick blanket of snow, and yet the bird is still quite accurate. How does it do this? Well, now I want to look at differences between food storing birds, so our Clark's nutcracker, and a non-food storing, non-corvid, so your pigeon. And why the heck am I using pigeons? Well, many of you might know that we know quite a bit about spatial cognition in pigeons. And even though their spatial abilities or their spatial cognitive abilities are different from nutcrackers or used to solve a different problem, they're still highly spatial birds. So some of you may know about pigeon homing, and this is the ability of pigeons to be released typically en masse from a location where they potentially have never been before, and they can fly back to their home loft. And we know that they use different kinds of spatial cues, magnetic cues, polar cues, and visual cues in engaging in these tasks. So I wanted to look at two different kinds of spatial mechanisms, if you will, or spatial problems, to look at how they use spatial cues in a local sense, whether they use them in similar ways or different. So this is a paradigm that was developed after Ken Chang's 1986 paper to look at whether rats encode local cues, featural items, and geometric information. So it's a fully enclosed rectangular arena, and at each corner of the rectangular arena, there's an object that's unique according to its color and its shape in my experiment. And in front of each of these features, there's an identical tin container. Only one of those tin containers contain reinforcement. So the bird has to learn which corner is the food hidden in. So in my example, I'm going to usually use the red object in this particular corner. But of course, I counterbalance that across corners with different birds. But we can also encode where that food reward is. So again, I said it's in this corner here. We can encode it according to geometric information. So if you, for a second, forget about the distinctive features in that rectangular environment and encode only the metric properties, so distance, direction, you can see that this corner has a long wall to the left and a short wall to the right. But the animals in these experiments are disoriented. So they're slowly spun in circles and taken on a random walk. So they can't use any kind of initial cueing like path integration. So they're disoriented. So they could be just as easily going in this direction, in which case this corner has a long wall to the left and a short wall to the right. So these two corners are identical. These rotational corners are identical according to geometric information. So I wanted to ask, do pigeons and nutcrackers encode featural information, and do they encode geometric information? And for the remainder of this part of my talk, I'm going to be using the schematic because I think it's a little bit easier to see than the actual pictures. And I'm going to be using the red location, or the one with the long wall to the left and the short wall to the right, as the rewarded location. And I'm going to ask whether these two species show differences in whether cues are reliable or unreliable. And I'm going to focus by talking on features. So one group of pigeons and one group of nutcrackers were trained in a situation where features were 100% reliable. The food was always located in the same location, so it's a reference memory task. And once they were accurately finding the food, we would give them test trials. A second group of birds, one group of nutcrackers, one group of pigeons, were trained with unreliable features. So what we did is we first trained the birds in a situation where there were no distinctive informative featural cues. There were featural cues, but they were identical in each corner, so they're pretty useless. So that the only thing that the birds could use is the geometric shape of the environment. They could only get to 50% correct according to the two geometrically correct corners. And then we retrained them in the same situation now with features 100%. So in this situation, geometry is actually more informative than features. Features are unreliable. And then we asked, do the birds encode the geometry of the environment? So now we take away all the informative featural cues and just leave the birds with the geometric properties of the environment. If they had encoded geometry, they should divide their choices between the two geometrically correct corners. And then we asked, well, what happens if we put features and geometry in conflict? So to do this, we took each one of these features and we moved it one position clockwise. So now you'll see the red feature is over in this corner and the blue one is down here. So if the birds are weighing featural information more importantly, they should follow that feature into a geometrically incorrect corner. If they're weighing geometry more heavily, then they should continue to go to the two geometrically correct corners, even though they contain featurally incorrect information in those corners. Okay, so let's look at the groups that were trained with reliable features to see what they did when we took those features away. And you'll see that both groups, both pigeons and nutcrackers, show this very strong encoding of geometry. Recall, they don't need to encode geometry in this situation. Features are 100% reliable, yet they still incidentally encoded geometry. So the majority of their choices are to the two geometrically correct corners. So we know they use geometry. What happens when we put these cues in conflict? So again, we take the feature and we put it in a geometric corner and vice versa. And we see that both pigeons and nutcrackers use features more than geometry, but there's a species difference. Pigeons only go to the correct corner according to features 76% of the time, but nutcrackers show a whopping 95% of the time to the correct features. So when features are reliable, the nutcrackers show a stronger weighing of features compared to pigeons. Okay, so we know that both of them can use features. We know that they show an incidental encoding of geometry, and that we see these species differences when the cues are in conflict in a feature reliable situation. What happens in the unreliable situation? So the birds are first trained with geometry and then later retrained with features. Perhaps not surprisingly, they encode geometry. Well, we trained them to do so, so it wasn't that surprising. And we see this strong control by geometry for both groups. But when we look at the cue conflict situation, we again see a species difference, and I'm going to walk you through this. So the pigeons show half of their responses, 51% of their responses to the correct feature. And then the other half of their responses are to the correct geometry. So they're dividing their choices between features and geometry quite equally. The nutcrackers are showing a different pattern. They're showing the majority of their choices are to these two geometrically correct corners. So now when the birds are trained that features are unreliable, the nutcrackers are switching to the more reliable cue, the geometry in this case. So we see that the birds are using these cues differently. So they both encode features, they both show an incidental learning of geometry, and they can learn to encode it when we train them to, but they weigh features and geometry differently depending on the reliability of these cues. So the general summary then is food storing birds, like nutcrackers, may be more influenced by spatial cue reliability than non-food storing birds. And of course, there are many differences between Clark's nutcrackers and pigeons, other than the fact that one stores food and one doesn't. But again, I think this is an interesting line of research to try to understand how these birds use their spatial cues within their environment that allows food storing birds to be so accurate. Okay, now I'm going to switch gears to my third theme for the talk. And this was asking about abstract concept learning. So do food storing birds have an ability to form abstract concepts, or sometimes referred to as relational concepts? So this work on abstract concept learning in non-human animals has been conducted in large part by these two individuals, Jeff Katz and Tony Wright. And they have looked at whether rhesus monkeys, capuchin monkeys, and pigeons are able to learn abstract concepts. And they use what's called a same different task. And I'm going to walk you through this task in a couple of steps. First of all, hopefully you can see these are just to show you what the apparatus looks like. So this is, in my case, this is my lab, so there's a Nutcracker in there. But this is an operant chamber, so it's a fully enclosed chamber. And at the front of the chamber, there's a computer monitor. And the birds can, or the primates can sit and look at what's being displayed on that computer monitor. And around that computer monitor is an infrared touch frame, such that if you break the beams, the computer will tell you where the X and Y coordinate is of that beam break. So you can liken it to when you go to the ATM and you touch that you want to take money out of your checking and not your savings. So these birds are looking at visual stimuli and they're making decisions by pecking on the screen if they're a bird or touching the screen for the primate studies. Now this is a same different task. So this task involves showing birds same trials and different trials. What do I mean by that? Well, let me show you an example of a same trial. So the bird's looking at the computer monitor and an image comes up. We call this the sample. And the bird is required to peck at that sample image a fixed number of times. We typically use 20, and the primates touch that image 20 times. Once they've fulfilled that requirement, a comparison image comes up. Alongside it, a black square. On a same trial, the animal is taught if you touch the comparison image, you will get a reward, juice for primates, food for birds. If you touch the black square, the lights in the chamber will go out for a short period of time. This is different from different trials. On a different trial, the image comes up, the primate touches it, the pigeons and nutcrackers will peck it for a certain number of times. Once that fixed ratio is completed, a second comparison image comes up, and that image is different from the sample. If it's a different image, what the animals are trained to do is to touch or peck at the black square. If they do that, they get a reward. If they touch the comparison image, the lights in the chamber go out for a short period of time. We adopted this exact same procedures that Tony Wright and Jeff Katz have used in many studies now, looking at abstract concept learning in monkeys and pigeons. So in our case, birds are going to be trained on this 2 items. So there's a sample and a comparison on this same different task. The sessions are 100 trials long. 50 of those are same trials and 50 of those are different trials. And they have to reach typically an 80% learning criteria. Once they hit that criteria, we give them transfer sessions. So they get 6 transfer sessions. And in a daily session, they still get 100 trials, but 10 of them are with stimuli they have never seen before. So they're novel stimuli, and they're intermixed among 90 training images, the ones that they have seen before. And we do what's called a set size expansion. So first, the animals are trained only with 8 images. That's their entire image set, just 8 images. Once they learn those images to the learning criteria, they're given the transfer. After the 6 transfer sessions, they're now trained, we're skipping 16 for reasons you can ask me later, to 32. So we expand the set size. Now there's 32 different images that they see, they're trained, they hit criteria, they get the transfer images, and then they go up to 64 and so on and so forth. So we double the set size, we transfer test, and we repeat. So what this allows us to do is try to understand how the animals are solving this task. So they can solve the task by using item-specific learning. So that's really learning about these 2 pairs of objects, or these 2 pairs of images, if you will, and the reinforcement outcome, what behavior they need to do in order to get the reinforcement. Or they can learn about them in relative terms, so they can relationally learn them. These 2 items, these 2 images provide the same information, or on different trials, they provide different information, right? So they don't have to learn about the exact exemplars. And just to try to show you what the difference is between these two ways of encoding, you can think about the relational. It's same or it's different. For us, that's pretty easy to think about. Well, what about this item-specific learning? Well, the animals get trained with 8 images. Here's the 8 images that we use. If they're learning item-specific, they have to learn about that item matched with itself and that item matched with every other different item. So that leads to 64 unique exemplars that they have to learn about. But it gets even more crazy. So at 16, they have to learn 256 unique exemplars. At 32 items training, they have to learn over 1,000. At 64, they have to learn over 4,000 exemplars. And we can keep going up to over 1 million. So these are very different outcomes. So if the animals are solving the task, if they're using this item-specific learning, they should fail when we give them new objects or new images, right? Because they've never seen those before, so they have no item-specific memory content for those novel images. If they're showing relational learning, they should pass this transfer with novel images because they're encoding it relationally. They're either same or they're different. So it doesn't matter the exact content of that picture, but they can pass in 2 ways. One we'll call partial transfer, and that's just that they're better than chance, right? So there's 2 responses that you can make. So chance is 50%, or they can show what we call full transfer. And this is transfer to those novel images or performance on those novel images that doesn't differ from how they responded during training. Question?
Yeah, I'm sorry to interrupt. I just wanted you to repeat what do they actually do in the transfer task?
Okay, so they learn a set of training images that
with
start
same different judgments.
Yeah, in the same different trials, and they get 8 pictures, 8 unique pictures. Mm-hmm. Once they hit a training criteria of 80%, so they're 80% correct at saying, yep, those 2 pictures are the same, or those 2 pictures are different. Then we give them these transfer tests, and we give them 6 sessions, so 6 days of transfer tests. And what happens during a transfer test is they get a picture that they've never seen before. So in that example, I showed you the apples, and I always forget what they are. There's apples and water and city scene and things like that. Well, I might throw in a picture of a monkey. Well, they've never seen that before. So they can't go back and look at the item specific and say, oh, right, when I saw monkey and monkey, I touch monkey. And if I see monkey and dog, I touch black square, because I've never seen them before. Mm-hmm. So if they can pass that test, if they show better than 50%, or if they show no difference than training, they had to have encoded it relationally. They have to encode it as, oh, those 2 pictures of the monkey are the same.
So it's, again, a same different judgment, only with new items.
Absolutely correct. Absolutely correct. Yeah. Yeah. Okay. So this is our 2 criteria that we're going to be looking at to say whether the birds and primates from the previous work are showing abstract concept learning. All right. Just to kind of outline how I'm going to be talking about this. This is the original work from Katz and Wright 2006, which we replicated their procedures. And I mean, we replicated them. We looked at visual angle. We looked at distance from the monitor to the animal. We used the exact same travel slides that they use. So we did everything we could to try to replicate this as close as possible. And I'm just going to blow this up a bit so you can see it more carefully. So this is the percent correct along the y-axis. This is that increasing set size that I talked to you about on the x-axis. And what we're looking at here is if it's a filled symbol, it's baseline. If it's an open symbol, it's that transfer performance. And what you can see is the animals learn the task quite well. But when you give them the very first set of those transfer images, those novel images that they've never seen before, performance plummets. And it really is not significantly different from chance. So they don't show any evidence in this case of abstract concept learning using relational learning, right? So that's what we're looking at here. So then we can move on and see, okay, well, what happens with 32 items? Well, they're better than 50%, but they don't show this full concept learning that we're looking for here, which is 80%, because that was our training criteria. And it's not until 256 exemplars. So the pigeons have to have experience with 250 images in order to go, aha, or maybe they don't go, aha, but to go, aha, this is same, different. It's a relational concept that needs to be learned. The capuchin monkeys show it earlier with 128 exemplars. And the rhesus monkeys also show it at 128 exemplars. Okay, and so the researchers, Katz and Wright and their colleagues, say that same, different concept differs in quantity but not in quality across the species. So the species that they studied all learn to use a relational concept, but they differ in how they get there. So we recruited John Magnotti, Alize, you met her before earlier in my talk, my PhD student, and Kevin Leonard, research student in my lab, amazing student, glad to have him on board. And we were looking at whether corvids are able to learn this abstract concept learning. Okay, exact same procedures as Wright and Katz, as best as we possibly could. 7 wild-caught Clark's nutcrackers, and here's the same picture that I showed you before of the nutcracker performing in this task. Exactly the same, 80% criteria, 100 trials per session, 50 same, 50 different, transfer tests to these novel stimuli. We use this set size expansion paradigm. And let's look at what they found, what we found, but let's break it down according to those set sizes. So the very first transfer they get is after they've only had learning with 8 items. And remember, pigeons, rhesus monkeys, and capuchin monkeys were at chance. Here's our individual nutcrackers. So here's percent correct on the Y, and our individual birds, our birds like the bats also have names, on the X-axis. And what you can see is 2 birds show full transfer. So just after 8 items, 2 individuals show full transfer. 4 individuals show partial transfer. So they're better than 50, but less than 80. And then 3 birds don't really know what's going on. If we look at this in terms of group data, so that was individual data. If we now look at the group data, here's our nutcrackers in blue, percent correct on the y-axis, and we can compare them to rhesus monkeys in orange, capuchins in yellow, and pigeons in green. And you can see that there's an amazing species difference after being trained with 8 items. And this is just to put those birds' data as a group in with the other species that have been conducted. And you'll see that as a group now, not on an individual basis, but as a group, the Clark's nutcrackers, rhesus monkeys, and capuchins show this full transfer at 128. And just to remind you, the pigeons were at 256. So our corvids, our nutcrackers, are showing no differences between these non-human primates. So then, of course, the question was, well, is this ability due to cache reliance? So when nutcrackers make their caches, one of the things that have been found over the years is that they encode these caches using relational metrics, whereas pigeons tend not to. They can, but they tend not to use relational metrics. So we were wondering, well, are nutcrackers really good at that task because they have this tendency to encode things relationally? So is it not just space that they're encoding relationally, but maybe they're encoding these concept tasks relationally as well, as we have seen. So we decided to take another corvid, but one that caches less. So they still cache, but they don't cache as often, as many items, and they don't retain those spatial locations in memory for as long. So this is the black-billed magpie. And I'm not going to make you go through the procedures again. It was exactly the same. Here's our results. On the y-axis, again, our percent correct in our individual birds. We had more birds this time on the x-axis. And, whoops, sorry, and this is after that 8-item learning. So again, just 8 items, and we can see that 3 of our birds are showing full transfer, 4 of them are showing partial transfer, and 3 of them are not transferring at all. So they're very much like the Clark's Nutcrackers in that individuals were able to transfer after only learning about 8 exemplars. And we can add them to the group graph. So when we look at the magpies as a group now instead of individually, we can see that they're no different in performance to the Clark's Nutcrackers, but superior to the rhesus monkeys, capuchins, and pigeons. And we can then look at their set size expansion to see when, as a group, they reach relational concept learning. And they're no different from the Nutcrackers. They're the same as the rhesus monkeys and the capuchin monkeys. Okay, so to conclude this section, we know that Clark's Nutcrackers and black-billed magpies show remarkable abstract concept learning. And it's likely not due to cache reliance. Not saying that it's not related to caching, because of course both of these were caching corvids, but it's likely not related to how long they hold these caches in memory or how many caches they make. And that at the individual level, they really show this incredible 8-item transfer. So they can show this relational concept learning after 8 items. And as a group, their transfer performance doesn't differ from the 2 non-human species studied in this particular task. So where are we overall? I hope I've convinced you, or at least made you think a little bit more, that birds indeed are capable of what we call complex cognitive feats, or higher order, higher level cognition, some people call it. And that birds, food storing birds, are encoding something about who's watching them. So we've looked at this in terms of social cognition and where are the caches located, and that's the talks on spatial cognition. And when we look at social cognition, we see that the degree of sociality may be important, and I think future studies can really try to differentiate between, is it really social cognition? And we see these differences in cache protection strategies in Clark's Nutcrackers and in Pinyon jays. When I told you about spatial cognition, I told you that food storing birds, in this case represented by the Clark's Nutcracker, and non-caching birds represented by the pigeons, both encode features and geometry, but maybe how they rely on these cues differ depending on Nutcrackers using cues that are more reliable. Then we switch to abstract concept learning, and we use the exact same methodology as best as we can to address these questions of whether birds are capable of abstract concept learning. And we found that these 2 species of corvids show performance that really rivals non-human primates. So it'll be so exciting to start looking at other corvids that cache or ones that don't cache to try to tweak out the conclusions to this last area of research. So I'd like to end by thanking many people. These are ones that I just want to point out in particular, the students whose really this was their research projects, Dawson, Kevin, and Alize, and then my many collaborators that helped with these projects, Ken Chang, Al Camel, Jeff Katz, John Magnotti, Marcia Spetch, and Anthony Wright. And of course, thanks to all of you for listening to my talk, and I'm happy to answer any questions if there's time.
Well, many thanks for a wonderful talk, very clearly presented, impressive amount of work. However, we won't have questions now, so I'm told by the chair of our meeting that actually the questions will be delayed. We will first have a brief presentation by Mélanie Guigueno, and after that there will be time for questions till time is over, but then more questions can be asked during the discussion session later on today. So first of all, I'd like to give the floor to Mélanie, who is actually working at McGill University, also here in in Montreal, and she will be updating us on cowbirds and brood parasites and how they select how females select hosts. So this will be a brief presentation, about 5 minutes, and after that there will be time for questions, I hope.
Thank you very much. Okay. All right, hi everyone. So it's my pleasure today to talk about these wonderful birds. These are brown-headed cowbirds. And I'll just show how these birds, but specifically the one to your left here, is or can be a successful nest thief. Can everybody hear me correctly? It's loud enough? Good. Okay. But first, I'd like to acknowledge Debby Kelly's fabulous talk on specifically cognition, more broadly cognition in food-storing birds, which was split into 3 themes: social cognition, spatial cognition, and abstract concept learning. So what I'd like to focus on is spatial cognition. Okay, so I'll take to focus on one of her themes. Now, instead of comparing species, as did Debby, I like to look at within species differences, okay, more specifically sex differences. So in my research, I've used brown-headed cowbirds, and well, why, may you ask? Is this a good subject for studying spatial memory? Well, many of you may know that brown-headed cowbirds are obligate brood parasites, right? So they rely solely on other species to raise their young. So they rely on about 100 different, well, approximately 150 species, the exact number is not quite known, to raise their young. And we can find cowbirds and their hosts across North America. So we see the distribution map here. What's interesting about brown-headed cowbirds is that it is only the females that search for, parasitize before sunrise when it's dark, and revisit host nests, okay? So males do not help females. So there's quite a strong sex difference in essentially demands on females relative to males when it comes to spatial memory, okay? Now, just before proceeding with my research, I'd just like to emphasize the fact, too, that cowbirds are quite vilified, right? They parasitize hosts, some of which are endangered species. They reduce the reproductive success of many hosts. So another reason I think is quite interesting to me to study cowbirds is that by understanding their cognition, we begin to really appreciate these fascinating birds. All right, so one question I'd just like to address today, and I wanted to address during my PhD research, was do female cowbirds have spatial memory specialized for brood parasitism? And we initially predicted that females would outperform males on spatial memory tasks, so on 2 vastly different tasks, as we see frequently in the food-storing birds. So in food-storing birds, we see that food-storing species outperform non-food-storing species on a variety of spatial memory tasks. So we want to see if this was the case between males and female cowbirds. So briefly, we had an allocentric task in which birds had to move through their environment and remember a location for 24 hours. So this resembled more what female cowbirds have to do in the wild. They have to remember a host nest location and return to the host nest the next day, okay, and make multiple visits to that nest over several days. Also, I like to emphasize that they will lay up to 50 eggs a season, so they have to constantly update their spatial memory information. And finally, we looked at an egocentric task using touchscreens. So this was a stationary task in which birds had to remember location for just a few seconds. So relative to the other tasks, this task resembled less host nest revisits, right? When female cowbirds visit host nests and revisit them. So I just have a video here just to show the results. So essentially, the cowbirds were introduced to this apparatus. We had 25 cups, one of which was baited. And then 24 hours later, the cowbirds returned to the apparatus. All the cups were covered with paper. We had trained the cowbirds to remove the papers, and we counted the number of errors. And also we measured the paths that the cowbirds took to the baited cup. So what did we find? This is an example of a female returning to the apparatus 24 hours later. And then there we go. She finds the baited cup, no problem, no errors, and she went directly to the baited cup. Okay, what about a male? So see, this is an example of what males typically did. So one error there, another error there. Spends more time and goes in circles a few times. So we'll save him from further embarrassment and move on, but essentially he makes a few more errors before finding the baited cup. So in summary, females made fewer errors and also took a more direct path to the baited cup. So path tortuosity was simply the path length that the birds took to the baited cup divided by the shortest possible path. So a smaller number indicates better performance. Okay, so on this allocentric task that resembled what cowbirds do in the wild, females outperform males. Now, briefly, I just have one slide on this task. We also use our touchscreen apparatuses. So similar to Debby Kelly's setup, we place cowbirds in operant conditioning chambers with infrared touchscreens. And we showed shapes on the screen. And essentially, the cowbirds had to remember a location of a shape for a few seconds, 15 to 60 seconds. So that retention interval is quite a bit shorter than our allocentric task. So we found with this task, and especially for the shorter retention intervals, males outperform females. All right, so I'd just like to summarize and conclude. So on our allocentric task, females made fewer errors and took more direct paths than males. So females were better on this task. But on the egocentric task, actually males seem to outperform females. And this was especially true at the shorter retention intervals. So retention intervals that were closer to the 15, 20, 30 seconds. All right. So this suggests that females will outperform males and have better spatial memory than males, but it's just a specialized form of spatial memory. It's not necessarily generally, they're not better than males on spatial memory in general. All right, so they're better than males on ecologically relevant tasks. Okay, so the tasks do seem to matter. All right, so in conclusion, I just want to mention that our results so far are consistent with the hypothesis that spatial memory in female cowbirds is specialized for brood parasitism. In the future, I'd like to run these tasks on different species of brood parasites and also compare them to non-brood parasitic species. And that's it for now. Thank you very much for your attention. I'd be happy to answer your questions.
Thanks very much for a very brief but very much to the point presentation here. So now we do have actually some 15 minutes or more for discussion. I think it would be fair to first give the floor to Debby to answer some questions. I noticed that there were some people behind the microphones earlier on. So please, Debby, if you can come over here. And both Mélanie and Debby will be present at this afternoon panel session. So if you have questions for any of them, more opportunity for questions later on. So in the meantime, two people behind the microphones, first over there.
Yeah, hi, thank you. Regarding the abstract learning concept for the same different task, when they were used on primate mainly, they also had a third option, which is to to just pass the difficult task, and that is what they call the uncertainty monitoring, and that's how they believe that primates know that they don't know. Do you think that the same thing happened in bird? Like, can they monitor their uncertainty?
Well, that's an excellent empirical question.
Could you please use the microphone?
That's an excellent empirical question that we haven't asked in these birds. So that's a really neat task, and I like the possibility of being able to look at uncertainty and whether the birds can engage, think about how certain they are in order to solve this task. We didn't do that, and the study that we replicated by Katz and Wright didn't do that either. So we really tried to stick to replicating. I don't know. It's a great question, and of course I can pontificate and think about what I think my special Clark's Nutcrackers would do, or black-billed magpies. But I'd rather leave that and do the study.
Okay, thank you.
Great question.
Okay, we switch to the other side.
Yeah.
Art.
Do nutcrackers know they're thieves?
Do nutcrackers know they're thieves? That's an excellent question. The initial results from our study would probably suggest not, but I have an honors student that just finished working in my lab, and what she was interested in was understanding whether birds, and she focused primarily on pinyon jays, whether they would know or could they possibly learn that two pinyon jays may respond differently. So she referred to it as the friend and enemy task, and they're not friends nor enemies, but one would steal the caches and one wouldn't. And her results were complex, but they don't suggest that the pinyon jays in this study were able to quickly learn that one individual was a thief, whereas another individual was
we
not.
know that ravens can do that.
Right.
But let me just sort of rephrase the question. So here we have a nutcracker, and it knows it's a thief. And it's also quite sensitive to non-thievery when it's an inanimate object. It's not worried about that in any way. But it is worried about a pinyon jay that could possibly be a thief, in fact, probably is. Let's suppose that we put another species of bird in there as the observer that is known not to be a thief. Mm-hmm. Ecologically speaking, the nutcracker doesn't have to worry about whether this bird knows whether. So what happens now?
Yeah,
Do they treat it as an inanimate object, or do they worry about this could be a thief?
Right. That's an excellent empirical question as well. And we're slowly chipping away at that by, so in this honors student's thesis, one of the things she also did was have an inanimate object, but that inanimate object was a taxidermy bird. All right. So yeah, it doesn't answer your question, but I think we need to make sure that we have conditions that we can control for the different variables that could be accounting for behavior, right? So if they treat this taxidermy bird the same as an inanimate object, we've taken one more step, right? Because there are things that correlate with thieving, right? There's approaching the tray, there's digging in the tray. So all of those behavioral responses need to be carefully controlled for. So I think it's an excellent question, and I think we're doing our best to try to get at an answer to that. From the lab, I couldn't tell you an answer to your question immediately right now. Yeah, but it is one we're working on.
Okay, next question over there.
Hi, thank you very much for the talk. My question was, because we saw earlier that environment has an impact on the behavior of the animals. Mm-hmm. So I was wondering if the birds that you take for your experiments were in the wild before, or they were raised in laboratories, and if so, do you think that it might have an influence on the results?
Oh, that's an excellent question. Yeah, so all the birds, so the Clark's nutcrackers and the pinyon jays were wild caught as adults and brought into the lab. The black-billed magpies I talked to you about were taken from the wild as pre-fledglings, and they were hand-reared in the lab. And the pigeons were in the wild in that they were previous racing pigeons. So as I mentioned, pigeons can engage in this homing behavior. So they had experience in the wild. So all of our birds had different degrees, I guess you could say, of natural experience. Do I think that makes a difference? We haven't directly tested that in those species. Many years ago in my PhD, my supervisor and I looked at whether pigeons with racing experience or pigeons without racing experience, so ones that were reared in the laboratory basically, showed differences in their ability or in how they use spatial cognition from images. So we showed them images, and somewhere in those images, if they pecked on the right location, they would get a food reward. And so we showed them images across a campus, from up close, from far away. So they really had to use the spatial relationship among within the image in order to determine where the food reward was associated. And we didn't see huge differences. Now, that's just one species, and I try not to generalize to other ones based on one. So I think it's a great question. It's just one that we haven't looked at in our studies in particular, other than the pigeon work that we've done.
Thank you. Next question.
Yes. Hi, thanks again for the talk. It was very interesting. I wanted to know, when it comes to what you call concept learning, or the category learnings that these birds do. So in the transfer task, what kind of categories do you show birds? How can you rule out that they're not performing on the task based on similarity? So for example, if you show a shoe and you show an apple, they clearly see that it's very different. So then they have to peck on the black square. So how can you, let's say, make sure that they're actually doing this based in the category, or what you're calling the concept, and not only in the perceptual input?
Yeah, that's an excellent question. And that's a question that Tony Wright and Jeff Katz have spent many hours worrying about. And they have several publications where they've very carefully gone through and tried to control for those kinds of alternative explanations. If they were here, they would provide you with a much more thorough answer to that question. But you can do things like not have the transfer tests. You can do things like have some birds learn 8 and then 16 and then 32, and looked at transfer after. You can look at transfer throughout. You can look and see how similar the images are on, a pixel by pixel level. So they've done a lot of that previous work. So we didn't jump in by starting back at square one, if you will, and seeing what Nutcrackers do. We adopted their procedures where they've kindly ironed out those kinds of problems, entropy, those kinds of things, to focus on the main question of whether Nutcrackers would show this abstract concept learning. It's a very important question, and I would really encourage you to have a look at some of Jeff Katz and Tony Wright's earlier work. 2006 paper would be a lovely place to start, in order to see the effort that has gone into really making sure that it can't be solved by these lower level perceptual solutions. Yeah.
Okay, thank you.
Next question.
Well, we've had 2 presenters today talking about animal personalities, and particularly with Debbie's presentation, what I saw is as much within species variation as across species variation. So my question is, maybe for both of you, are we looking at the wrong level when we're lumping all species members together? And I'd be happy to have both of you come up and answer that.
Should I take it first?
Okay, Jim, would you have anything to say to that? Jim has nothing to say, but perhaps Debbie can say something about it.
My answer to that is probably not what you want to hear, but I don't think that we're asking the wrong question. I think we're asking different questions, right? One is more a question of individual differences and what are individuals within a species capable of, if you will. Whereas the other question is more along looking at those groups, which is why I showed you the individual data and then the group data. So if we looked at, let's just take the Nutcrackers on the concept learning, it looks as a group that they didn't develop or weren't successful at full transfer until 128 items. But if we look at an individual basis, some of those individuals solved the task relationally much earlier. So I think we're asking different, those 2 approaches are answering different kinds of questions. And I think that both of them are important.
Well, the problem is that if you have particular individuals of a species that are very successful or who very unsuccessful, you might say the species can or cannot do the task. And I think you could say, well, we have a few very, very successful individuals, so the species can do the task. But I think that gets away from the problem of some species can do it and some species can't, because
Right.
I don't think at that point we know what's going on.
I mean, it's analogous to humans. So we do a lot of spatial cognition research in humans as well. And we can say humans can solve X or they can't. Well, that's like comparing me and Einstein. Of course, he's going to be able to solve questions that I can't. Maybe I can solve some that he can't. But we see these individual differences in humans. I think, we tend, because they're smaller numbers, there's so many things that you'd have to contemplate, but we tend to say species can do things and species can't do things. But I mean, it's similar to saying males are good at spatial memory and women are not. Well, that's baloney. It's not true, right? There are different individuals that are good at certain tasks and different individuals that are poor at tasks. So I think we're asking different well, not we, but as a field, we're asking different kinds of questions. We're answering different kinds of questions. Can individuals solve these tasks? If we look at the bigger group, how does the group respond? We do the same thing in human cognition.
Yeah, it just worries me when we have so much overlap, that's all.
Okay.
Yep.
Hi. Nutcrackers live in pairs, if I understood correctly.
They're more territorial. So they don't, they tend not to live in these big groups. So nutcrackers will come into a feeding site, gather pine nuts and then disperse. Whereas the pinyon jays that I talked to you about live more in larger groups. So they can be in a flock up to about 100 individuals.
Okay, so they don't associate in pairs, as in they don't bond in pairs?
Nutcrackers
Yes.
do. So they will form pairs and mate and rear offspring in the
Together, okay.
Yeah.
So do their thieving behavior differ with respect to partners as opposed to non-partners?
That's a awesome question, and I don't know the answer to that because we haven't done those studies. So it would be, as we were talking about earlier, in terms of recognition of individuals and thieves versus non-thieves. That's another future question that certainly needs to be tackled. Are relatives different than non-relatives? Are mates different than a relative, for instance? Those are all great questions. I'm sorry I can't answer them because we haven't done those empirical studies yet.
Thank you.
Yeah.
Hi, thanks. I was just wondering, do you know of any data on human children on the transfer task?
I should. It has been done in human children. The developmental area is not my area. But this same different discrimination has been shown in children. It's been shown in bees, more recently in chicks, but the procedures are different. So what we can compare and contrast across them really is dependent upon the procedures that are used, I would argue. So I'm not going to go out on a limb and say, nutcrackers are, better than young children, or young children are better than nutcrackers, because that hasn't been done.
Okay, thanks.
Yeah, great question.
All right. I think, I'm afraid, in view of the time, we should finish the discussion now. That was also a great question leading up to the bees, about which we will hear more tomorrow. So thank you all for being present, and thank the speakers again for their very nice presentations.